Hello, beautiful souls. Welcome to this quiet space, a place where minds slow down and hearts rest. Before we begin this gentle, sleepy journey, just a small moment of support. If this meditation brings you calm, comfort, or clarity, please subscribe to the channel, gently tap the like button, and leave a comment to let us know you are here. Your support helps us continue creating these peaceful spaces for your mind, for your heart. Now, there is nothing else you need to do, nothing to think about. Let's begin. Close your eyes now and slowly settle into a comfortable position. Take a gentle breath in and slowly breathe out. Let your shoulders relax, let your jaw loosen, and allow your whole body to feel heavy. When scientists observe the natural world, they do so without applying human concepts of kindness or cruelty, and instead, they measure patterns of energy flow, resource distribution, and statistical frequency of various biological events. The phrase, "the dark side of nature," is itself a human construction, a linguistic framework that attempts to categorize certain observable phenomena that humans find discomforting or difficult to reconcile with preferences for comfort and predictability. In biological terms, there is no darkness or lightness, no good or evil, only processes that occur with regularity according to physical and chemical principles that have operated for billions of years without pause or variation in their fundamental mechanisms. Nature, as an abstraction referring to all living and non-living systems on Earth, does not possess intention, preference, or awareness. It is not a conscious entity that makes choices or determines outcomes based on desired results. When we observe what appears to be suffering, competition, or destruction in natural systems, we are observing the routine operation of thermodynamic principles, genetic expression, and ecological interactions that proceed according to probability distributions and energy gradients. The universe operates through forces and particles interacting in space and time, and biological organisms are arrangements of matter that temporarily maintain organization through constant energy input following patterns encoded in molecular structures we call genes. Predation is a term scientists use to describe the process by which one organism obtains energy and nutrients by consuming another living organism. This process is not an event in the narrative sense, but rather a continuous transfer of chemical energy from one form to another, similar to how water flows from higher to lower elevations due to gravitational potential. When a predator consumes prey, molecules that were organized in one particular arrangement are broken down through digestive processes and rearranged into different molecular configurations within the predator's body. The energy that was stored in chemical bonds within the prey organism becomes available to power cellular processes in the predator organism, and this transfer follows the laws of thermodynamics with no capacity for deviation or choice. From the perspective of energy flow through ecosystems, predation represents one pathway through which solar energy, originally captured by photosynthetic organisms, moves through various levels of biological organization. Plants convert light energy into chemical energy stored in glucose molecules, and this energy then becomes available to organisms that consume plants, and subsequently to organisms that consume those plant-eating organisms. At each transfer, a significant portion of energy is lost as heat, dissipating into the environment in accordance with the second law of thermodynamics, which describes the tendency of organized systems to gradually lose organization over time unless energy is continuously added. The act of one animal killing and consuming another animal occurs millions of times each day across the planet in oceans, forests, grasslands, deserts, and every other ecosystem type. Each of these events is statistically ordinary, predictable in aggregate, even if individual instances cannot be precisely forecasted. Lions on African savannas consume zebras and wildebeests with a frequency determined by lion population density, prey population density, lion energy requirements, hunting success rates, and numerous other measurable variables. None of these variables involve concepts of right or wrong, mercy or cruelty, but only quantities that can be measured and compared across time periods and geographic locations. Competition for resources is another fundamental process that shapes biological systems. Resources, in scientific terms, are materials or energy sources required for survival and reproduction, but available in quantities insufficient to meet the needs of all organisms simultaneously. These include nutrients such as nitrogen and phosphorus, water, suitable habitat space, mates for reproduction, and energy-rich food sources. When multiple organisms require access to the same limited resource, competition occurs, meaning that one organism's consumption of that resource reduces its availability to other organisms. This competition manifests in many forms, some involving direct interaction between organisms, and others occurring without any contact. When plants grow in close proximity, their root systems expand through soil in search of water and mineral nutrients. The presence of one plant's roots in a particular soil volume means those resources cannot be simultaneously accessed by another plant's roots in that same volume. The plants are not aware of competing, they are simply growing according to genetic programs that direct cellular division and expansion in patterns shaped by millions of years of natural selection. Natural selection itself is not a purposeful process, but rather a statistical description of which genetic variants tend to increase in frequency over generations. Animals compete for food resources, for territory, for access to mates, and for various other necessities. When food is scarce, which is a common condition in nature rather than an exceptional one, organisms that are more efficient at locating and consuming food, or more capable of defending food sources from others, will obtain more energy. This energy differential affects survival probability and reproductive output. There is no fairness consideration in this process, no mechanism that ensures equal distribution, only the mathematical reality that finite quantities, divided among multiple entities, results in some entities receiving more and others receiving less. Aggression is a behavior pattern that scientists observe in many animal species. It can be studied through neurological mechanisms, hormonal systems, and evolutionary contexts, without any reference to moral frameworks or emotional interpretations. Aggressive behaviors include physical attacks, threat displays, territorial defense, and various other actions that involve one organism attempting to harm or displace another organism. These behaviors are generated by neural circuits in the brain that respond to environmental stimuli and internal physiological states. They are mediated by chemical signals such as testosterone, serotonin, dopamine, and numerous other neurotransmitters and hormones. From an evolutionary perspective, aggressive behaviors persist in populations because they have statistical associations with survival and reproductive success in certain environmental contexts. An organism that successfully defends a territory containing food resources or suitable nesting sites will have access to those resources without competition. This increases the probability of surviving through periods of scarcity and successfully raising offspring. An organism that fails to defend such resources may experience reduced energy intake, increased exposure to predators or harsh weather, and decreased reproductive output. Over many generations, genetic variants that influence the likelihood or intensity of aggressive behavior in specific situations can increase or decrease in frequency depending on the average outcomes associated with those behaviors in the prevailing environmental conditions. The neural mechanisms underlying aggression involve specific brain regions, such as the hypothalamus and amygdala. These regions integrate sensory information about threats, resource availability, and social context. They send signals to motor control areas that coordinate physical movements involved in fighting or threat display. The entire process occurs through electrochemical signaling within and between neurons, with no requirement for conscious deliberation or moral evaluation. An animal responding aggressively to a territorial intruder is executing a program generated by its nervous system in response to particular patterns of stimulation. This occurs much like a computer executing code in response to input data. Parasitism represents another category of interaction between organisms. It is one in which one organism, the parasite, obtains nutrients and energy from another organism, the host. This typically causes some degree of harm to the host in the process. Parasites include viruses, bacteria, protozoa, fungi, worms, arthropods, and various other life forms, occupying nearly every taxonomic group. They live on or inside host organisms, consuming host tissues, diverting host nutrients to support parasite growth and reproduction. Sometimes, they manipulate host behavior or physiology in ways that benefit parasite transmission to new hosts. From the parasite's perspective, which is not a conscious perspective, but rather a description of how natural selection has shaped parasite traits, the host organism is an environment providing resources necessary for survival and reproduction. A tapeworm residing in an animal's intestine absorbs nutrients from partially digested food passing through the intestinal tract. It uses these nutrients to produce thousands or millions of eggs that exit the host's body in feces and potentially infect new hosts. The tapeworm possesses no awareness of causing harm, no capacity for empathy or restraint. Only genetic programs directing growth, reproduction, and survival according to patterns refined over countless generations of natural selection. Many parasite species have evolved remarkable specificity. They infect only particular host species or only particular tissues within those hosts. This specificity results from molecular interactions between parasite surface proteins and host cell receptors. It also results from immune system evasion mechanisms and various other biochemical and physiological adaptations. A malaria parasite can only reproduce inside red blood cells of particular vertebrate species. This is because its life cycle includes stages requiring the specific cellular environment those blood cells provide. This requirement is not a choice, but a biochemical constraint determined by the parasite's genetic makeup. The concept of suffering, so central to human ethical frameworks, does not appear in biological descriptions of nature, because suffering is a subjective experience requiring consciousness and the capacity for negative emotional states. While neuroscientists can identify brain structures and chemical signals associated with pain perception and distress responses in various animal species, particularly mammals and birds, with complex nervous systems, the question of whether these responses constitute suffering in a phenomenological sense remains philosophically debatable and scientifically difficult to assess. Pain responses serve clear biological functions as warning signals indicating tissue damage or dangerous situations requiring avoidance or escape. When an animal experiences tissue damage. Specialized sensory neurons, called nociceptors, detect mechanical stress, temperature extremes, or chemical signals associated with inflammation. These neurons transmit signals to the spinal cord and brain regions involved in processing threat information. This transmission triggers behavioral responses, such as withdrawing from the pain source, vocalizing, or entering a physiological stress state that mobilizes energy for escape or defense. The entire process can be understood in terms of neural circuitry and chemical signaling without requiring assumptions about subjective experience. Natural selection, the process through which genetic variants change in frequency across generations, operates without intention, foresight, or desired outcomes. It is a statistical description of patterns that emerge when organisms with different genetic makeups survive and reproduce at different rates. When environmental conditions change, organisms carrying genetic variants that happen to make them better suited to the new conditions tend to leave more surviving offspring than organisms lacking those variants. Consequently, those advantageous variants increase in frequency in subsequent generations. This process requires no planning, no consciousness, and no purpose beyond the mathematical relationship between traits and survival probability. The mechanism of natural selection can be illustrated through changes in population genetics over time in response to environmental pressures. Consider a population of insects living in a forest where most tree bark is light-colored. Insects with genetic variants producing darker body coloration are more visible to bird predators against this light background. Consequently, these darker insects are eaten at higher rates than lighter-colored insects. Over time, the frequency of genetic variants associated with lighter coloration increases in the population, not because the population is trying to become lighter or because natural selection prefers lightness, but simply because the mathematical relationship between coloration and predation risk produces a consistent directional change in allele frequencies. If the forest experiences a fire that darkens the tree bark, the same process operates in reverse. Darker insects now have lower predation risk, and their associated genetic variants increase in frequency. Natural selection does not remember the previous environmental conditions, does not plan for future changes, and does not optimize for any particular outcome, except in the narrow sense that organisms tend to become better matched to their immediate environment through this ongoing statistical process. The process is blind, automatic, and inevitable given the existence of heritable variation in traits affecting survival and reproduction. Scarcity of resources shapes ecosystems through continuous influence on population dynamics, community structure, and evolutionary trajectories. In any ecosystem, the total amount of available energy, water, nutrients, and suitable habitat is limited by physical constraints such as solar radiation input, precipitation patterns, soil chemistry, and geographic area. These limitations mean that biological populations cannot grow indefinitely, but instead are regulated by the carrying capacity of their environment. Carrying capacity is the maximum population size that available resources can sustainably support. When a population grows beyond its environment's carrying capacity, resource scarcity intensifies. This leads to increased competition, reduced individual body condition, decreased reproductive rates, and increased mortality rates. These density-dependent processes cause population size to decline back toward carrying capacity levels. When population size is below carrying capacity, resources are relatively abundant per individual. Competition is less intense, body condition improves, reproductive rates increase, and mortality rates decrease. This causes population size to increase back toward carrying capacity. This dynamic creates population fluctuations around carrying capacity rather than stable equilibrium states. Scarcity also drives competitive interactions between species occupying similar ecological niches. When two species require the same limiting resources, the species that is more efficient at acquiring and utilizing those resources will tend to increase in abundance. The less efficient species declines, potentially leading to local extinction. This process, called competitive exclusion, has been documented in numerous laboratory and field studies across many taxonomic groups. It occurs not through active exclusion or deliberate harm, but through the mathematical consequences of differential resource acquisition efficiency. Death is statistically common in nature, particularly among young organisms. Most species produce far more offspring than can possibly survive to reproductive maturity. The vast majority of these offspring die at various stages of development. This pattern results from the mathematical reality that if all offspring survived and reproduced at the same rate as their parents, populations would grow exponentially. This growth would continue until resource exhaustion caused mass mortality events. Instead, high offspring mortality maintains populations near carrying capacity through continuous attrition rather than catastrophic collapse. Consider a fish species that produces 10,000 eggs per reproductive event. If all these eggs developed into adult fish and reproduced at similar rates, the population would increase by a factor of 5,000 each generation. Within just a few generations, the population would exceed the physical space available in their habitat. Instead, the vast majority of eggs and juvenile fish die from predation, starvation, disease, and various other causes. Only a tiny fraction survives to adulthood. This mortality is distributed across the life cycle in patterns that vary by species and environmental conditions. Some eggs fail to develop properly due to genetic abnormalities or environmental stress during development. Some larvae are consumed by predators immediately after hatching. Some juveniles starve when they cannot find sufficient food. Some are outcompeted by siblings or other juveniles. Some fall victim to parasites or diseases. Some are killed by extreme weather events or habitat disturbance. The cumulative effect of all these mortality sources produces the observed pattern of very high juvenile mortality and very low survival to adulthood. Infant mortality serves population regulating functions by adjusting population size to match resource availability. When food is abundant and environmental conditions favorable, a higher percentage of offspring survive to maturity. This allows population growth. When conditions deteriorate, juvenile mortality increases, slowing or reversing population growth. This regulatory mechanism operates automatically through the combined effects of starvation, disease, predation, and other mortality sources. Their intensity varies with population density and environmental quality. Cannibalism, the consumption of individuals of one's own species, occurs in many animal taxa under various circumstances. It can be observed in insects, fish, amphibians, reptiles, birds, and mammals, including primates. The behavior can be studied through ecological and evolutionary perspectives without moral or emotional frameworks. Simply as one possible feeding strategy among many others available to organisms. In some species, cannibalism occurs opportunistically when individuals encounter vulnerable conspecifics, particularly eggs, larvae, or juveniles. In situations where these conspecifics represent available food sources, a female spider may consume a male after mating, obtaining nutrients that contribute to egg production. A lioness may kill and consume cubs sired by a male she has just displaced. This eliminates offspring carrying other males' genes and allows her to become reproductively receptive sooner. A tadpole may consume other tadpoles in a pond where food is scarce. This provides protein and energy that would otherwise go to competitors. From an evolutionary perspective, cannibalism can increase individual fitness under certain conditions. despite reducing population size. If food scarcity threatens starvation, consuming a conspecific provides energy that may prevent death, even though one member of the population dies in the process. If consuming unrelated juveniles allows an individual to produce more offspring of its own, natural selection may favor genes predisposing towards such behavior, even when it appears detrimental to the species. Evolution operates on individuals and their genetic variants, not on species or populations as cohesive units. Behaviors that benefit individual reproductive success can spread even when they seem detrimental to the group. Nature does not optimize for comfort, suffering reduction, or well-being, because nature is not an agent with goals or preferences. The physical and biological processes that constitute nature operate according to physical laws and statistical regularities, without any inherent direction toward pleasant or unpleasant states. Natural selection produces adaptations that increase survival and reproductive success in specific environments, not adaptations that increase comfort or happiness, except insofar as these might correlate with survival and reproduction. Many organisms spend substantial portions of their lives in states that humans would characterize as uncomfortable or stressful. Migrating birds fly thousands of kilometers with minimal rest, depleting fat reserves, and experiencing extreme physiological stress. Marine mammals dive to great depths, withstanding enormous pressure and operating near their physiological limits for oxygen storage. Desert animals endure severe heat and chronic water scarcity. Parasites live inside host organisms where immune systems constantly attack them. These conditions are ordinary rather than exceptional. They represent the normal state of existence for countless organisms. The reason organisms endure such conditions is that natural selection shapes traits based on reproductive success, not comfort. A bird that migrates successfully to breeding grounds may raise offspring even if migration is physiologically stressful. A bird that remains in comfortable conditions but fails to reach breeding grounds leaves no offspring. Its genetic variants are removed from the population. Over generations, this process results in populations dominated by individuals capable of tolerating the stress required for successful reproduction, regardless of how uncomfortable that stress might be. Disease represents ecological interactions between pathogenic organisms and their hosts. These interactions involve complex dynamics of infection, transmission, immune response, and population-level patterns of susceptibility and resistance. Pathogenic organisms include viruses, bacteria, fungi, protozoa, and multicellular parasites. They can multiply within or on host organisms, often causing physiological damage or dysfunction in the process. These organisms are not inherently evil or malicious. They are simply living things pursuing survival and reproduction. They do so through strategies that happen to involve using other organisms as resources. When a pathogenic bacterium infects a host, it begins reproducing rapidly if environmental conditions within the host are suitable. Bacterial reproduction proceeds through binary fission. One cell divides into two cells, which then divide into 4, then 8, and so on. This process results in exponential growth. Growth continues until limiting factors such as nutrient depletion, immune system responses, or toxic waste accumulation slow or stop the process. Some bacteria remain localized at infection sites. Others spread throughout the host body via the circulatory system or other pathways. Host organisms have evolved various immune system mechanisms for detecting and eliminating pathogens. These include physical barriers like skin and mucous membranes, chemical defenses like stomach acid and antimicrobial peptides, and cellular defenses involving white blood cells that can engulf and destroy bacteria or release chemicals that kill infected cells. The interaction between pathogen reproduction and immune system responses determines infection outcomes. Some infections are eliminated quickly. Others persist chronically. Still others overwhelm immune defenses and cause severe disease or death. Fear responses in animals serve clear biological functions related to threat detection and avoidance. When an animal perceives a potential danger, such as a predator, specialized neural circuits in the amygdala and other brain regions process this threat information. They trigger a coordinated physiological response. This response includes release of stress hormones like cortisol and adrenaline, increased heart rate and blood pressure, redirection of blood flow to muscles, inhibition of digestive processes, and heightened sensory awareness. These changes prepare the animal for rapid escape or defensive action. The capacity for fear responses is highly conserved across animal species. This is because individuals capable of rapidly detecting and responding to threats have higher survival probability than individuals lacking such capabilities. An antelope that detects a lion and flees immediately is more likely to survive than an antelope that fails to notice the predator or responds slowly. Over evolutionary time, natural selection has refined fear response systems. It has tuned them to detect relevant threats accurately, while minimizing false alarms that waste energy on unnecessary escape responses. The neural mechanisms underlying fear involve sensory processing regions that detect threat-relevant stimuli, such as sudden movements, looming objects, or specific sounds and odors associated with predators. These sensory regions send signals to the amygdala. The amygdala evaluates the potential threat level based on current context and past experience. If the amygdala determines a significant threat exists, it activates the hypothalamus and brainstem regions controlling the stress response. This entire process can occur in milliseconds, allowing very rapid reaction to danger. Violence in nature, meaning physical harm inflicted by one organism upon another, is not abnormal or exceptional, but rather a routine occurrence resulting from various ecological and evolutionary processes. Predation involves violence as predators kill prey. Territorial disputes involve violence as animals defend resource-holding positions. Mating competition involves violence as males fight for access to females, or as females resist unwanted mating attempts. Parent-offspring conflict can involve violence as parents enforce weaning, or as offspring compete with siblings. Each instance of violence can be understood through cost-benefit analysis. In terms of energy expenditure, injury risk, and potential gains in resources or reproductive opportunities. An animal deciding whether to fight an intruder over a territory performs, not consciously, but through evolved mechanisms and implicit calculation. It compares the value of the territory against the costs and risks of fighting. If the territory is highly valuable and the intruder appears weak, fighting may be favored. If the territory is marginal and the intruder appears formidable, retreat may be favored. These calculations are implemented through neural and hormonal mechanisms. They respond to environmental cues and internal physiological states. Testosterone levels, previous fighting experience, assessment of opponent size and condition, energy reserves, presence of mates or offspring, and numerous other factors all influence fighting decisions. They do so through their effects on neural activity in decision-making brain regions. The resulting behavior emerges from these interacting factors. Without requiring conscious reasoning or moral evaluation, extinction, the permanent loss of a species from Earth, is a common outcome of evolutionary and ecological processes. The fossil record indicates that more than 99% of all species that have ever existed are now extinct. This demonstrates that extinction is the eventual fate of nearly all species, given sufficient time. Extinction occurs when death rates exceed birth rates for a sustained period, until no individuals remain, or when genetic changes gradually transform a species into a different species through evolution. Various factors contribute to extinction risk. Small population sizes reduce genetic diversity. This makes populations more vulnerable to environmental changes and reduces ability to adapt evolutionarily. Habitat loss reduces the geographic area available for populations to occupy. Climate change alters environmental conditions faster than species can adapt. Introduction of invasive species creates new competitive or predatory pressures. Disease outbreaks can devastate populations lacking immunity. The combined effects of these and other factors determine extinction probability. From an ecosystem perspective, extinction removes one node from the network of species interactions. Predators that relied on the extinct species must shift to alternative prey or decline themselves. Prey that were controlled by the extinct predator may increase in abundance. Competitors of the extinct species may expand into newly available niche space. Parasites specialized on the extinct species disappear along with their host. Over time, these cascading effects propagate through the ecosystem, potentially causing additional extinctions or major community reorganization. Environmental stress, meaning conditions that challenge an organism's physiological tolerances or resource requirements, shapes behavior through multiple pathways. Acute stress, such as exposure to extreme temperature or sudden food scarcity, triggers immediate behavioral changes, like seeking shelter or increasing foraging effort. Chronic stress, such as long-term habitat degradation or persistent food limitation, can cause more fundamental behavioral modifications, including changes in reproductive strategy, social behavior, or habitat use patterns. The physiological basis of stress responses involves the hypothalamic pituitary adrenal axis. This system coordinates hormonal responses to stressful conditions. When stress is detected, the hypothalamus releases corticotropin-releasing hormone. This stimulates the pituitary gland to release adrenocorticotropic hormone, which in turn stimulates the adrenal glands to release cortisol and other glucocorticoid hormones. These hormones mobilize energy reserves, suppress immune function, alter cognitive processing, and modify behavior in ways that may help the organism cope with the stressor. Prolonged elevation of stress hormones can have various negative effects on organisms, including immune suppression, increasing disease susceptibility, reduced growth rates, decreased reproductive function, and altered brain structure, affecting cognitive abilities. These effects are not design flaws. They are consequences of physiological trade-offs. The stress response system evolved to handle acute challenges requiring rapid energy mobilization, not chronic, ongoing stress. The costs of prolonged stress hormone exposure represent unavoidable physiological constraints. The energy costs of survival are substantial for all organisms. Maintaining cellular function requires continuous ADP production through metabolic processes. Movement requires additional energy beyond baseline metabolic needs. Thermoregulation in endothermic animals requires enormous energy expenditure to maintain stable body temperature. Growth and tissue repair consume energy. Reproduction demands energy for gamete production, offspring provisioning, and parental care. Immune function uses energy to produce antibodies and maintain cellular defenses. These energy requirements mean that organisms must obtain sufficient food to cover all these costs, while also storing reserves for periods when food is unavailable. The balance between energy intake and energy expenditure determines body condition, which affects survival probability, disease resistance, and reproductive capacity. Organisms that fail to achieve positive energy balance lose body mass, deplete energy reserves, and eventually die from starvation or succumb to disease or predation due to weakened condition. Different activities have different energy costs that can be measured in metabolic rate increases. Active foraging may increase metabolic rate 3 to 5 times above resting levels. Rapid flight or running can increase metabolic rate 10 to 15 times above resting. Reproduction can increase average metabolic rate 20 to 30% over non-reproductive periods due to the costs of producing eggs or provisioning young. These costs must all be paid through food acquisition or the organism dies. Natural systems do not maintain balance or equilibrium, but rather exhibit continuous fluctuation around changing average conditions. The concept of balance of nature is a human construct that does not accurately describe ecological dynamics. Populations fluctuate in size from year to year due to weather variation, disease outbreaks, predator-prey cycles, and random demographic events. Community composition changes as species invade or go locally extinct. Ecosystem properties, like productivity and nutrient cycling, vary with climate and disturbance patterns. What appears to be stability in nature often represents fluctuation within bounds rather than true equilibrium. A forest may appear stable over decades, but actually experiences continuous turnover as individual trees die and are replaced, with species composition shifting gradually in response to climatic trends. A predator-prey system may appear balanced, but actually cycles regularly, with predator and prey abundances oscillating out of phase with each other. An ecosystem may appear resilient, but actually exists in one of several possible stable states and can flip to an alternative state if pushed beyond certain thresholds. The timescales of ecological change vary enormously depending on the process being considered. Individual organism behavior can change in seconds or minutes in response to immediate stimuli. Population size can change within a single generation due to reproduction or mortality events. Community composition may shift over decades as slow-growing species gradually outcompete others. Evolutionary adaptation requires multiple generations and may take hundreds or thousands of years for significant change. Soil formation and major geomorphological changes occur over thousands to millions of years. This range of timescales means that patterns appearing stable over short observation periods may be changing directionally over longer periods. A scientist observing a population for 3 years may see apparently stable abundance, while a century-long dataset for the same population might reveal a declining trend. A forest that appears unchanging over a human lifetime is experiencing gradual species turnover and compositional change, visible only in data spanning centuries. The relevant time scale depends on the question being asked and the processes being studied. Climate patterns vary over multiple timescales, from daily weather fluctuations to glacial interglacial cycles spanning tens of thousands of years. These variations impose continuously changing selection pressures on organisms and communities. Species ranges expand and contract as climate zones shift geographically. Community composition adjusts as species tolerances are exceeded or new suitable habitat becomes available. Extinctions occur when climate change outpaces species' ability to migrate or adapt. All these processes proceed without regard for ecosystem stability or species preservation. Natural systems continue operating without awareness, consciousness, or purpose beyond the immediate physical and chemical processes governing them. There is no overarching intelligence directing ecosystem function, no consciousness experiencing natural beauty or tragedy, no purpose beyond the continuation of physical law and the mathematical consequences of those laws applied to matter and energy. Rivers flow downhill because gravitational potential energy converts to kinetic energy, not because rivers desire to reach the ocean. Plants grow toward light because phototropins detect blue light wavelengths and initiate differential cell elongation, not because plants understand that light is necessary for photosynthesis. Evolution produces complex adaptations through accumulated small changes, each favored by natural selection because it slightly increased survival or reproductive success. Eyes did not evolve because organisms wanted to see, but because random mutations affecting light sensitivity provided small advantages in detecting food, predators, or mates. These advantages accumulated over millions of years and millions of small modifications. The apparently purposeful design of organisms is an illusion created by the cumulative effects of differential reproductive success over vast time periods. Ecological interactions proceed through chemical signals, physical contact, and energy exchanges without any requirement for understanding or intention. A bee visiting a flower obtains nectar for energy and incidentally transfers pollen between flowers, facilitating plant reproduction. The bee is not trying to help the plant reproduce, and the plant is not deliberately rewarding the bee. Instead, both organisms are executing behaviors shaped by natural selection, acting on their ancestors. Bees that efficiently located and extracted nectar left more offspring. Plants that produced attractive flowers and rewarding nectar received more pollination services and left more offspring. The current interaction is the result of this coevolutionary history. Nutrient cycling in ecosystems proceeds through decomposition, consumption, and biogeochemical transformations. Dead organic matter is broken down by bacteria and fungi that secrete enzymes to digest complex molecules into simpler compounds. These compounds are absorbed by decomposer organisms and incorporated into their biomass or released into soil and water. Plants absorb mineral nutrients from soil through root systems and incorporate them into plant tissues through biochemical synthesis pathways. Animals obtain nutrients by consuming plants or other animals and assimilating the compounds they need while excreting excess or unusable materials. This cycling continues indefinitely, with nutrients moving through organisms and environments in complex pathways determined by chemical properties and biological processes. Nitrogen cycles from atmosphere to soil to plants to animals to decomposers and back to atmosphere through nitrogen fixation, assimilation, ammonification, nitrification, and denitrification. Carbon cycles, from atmosphere, to plants, to animals, to decomposers, and back to atmosphere, through photosynthesis, consumption, respiration, and decomposition. These cycles operate through ordinary chemistry and biology, requiring no coordination or purpose beyond the molecular interactions involved. Weather and climate impose continuous influence on biological systems through temperature effects on metabolic rates and physiological function, through precipitation effects on water availability and habitat suitability, through wind effects on dispersal and mechanical stress, through solar radiation effects on energy input to ecosystems. Organisms respond to these physical conditions through physiological adjustments, behavioral modifications, and in some cases, evolutionary adaptation over longer time frames. Temperature directly affects the rate of chemical reactions, including all the biochemical reactions sustaining life. As temperature increases within an organism's tolerance range, metabolic rate increases approximately exponentially, roughly doubling with each 10 degrees Celsius increase. This relationship means that organisms living in warm environments have inherently higher metabolic rates and energy requirements than organisms in cool environments, all else being equal. It also means that seasonal or daily temperature variation causes corresponding variation in metabolic rate and energy needs. Water availability limits biological activity in many terrestrial environments. Cellular function requires water as a solvent for biochemical reactions and as a transport medium for nutrients and waste products. Photosynthesis requires water as a reactant. Terrestrial organisms continuously lose water through evaporation from respiratory surfaces and through excretion. This water must be replaced through drinking or eating water-containing foods or through metabolic water production. When water intake cannot match water loss, organisms become dehydrated. Cellular function deteriorates, and eventually, death occurs. Seasonal patterns in temperature and precipitation create cycles of favorable and unfavorable conditions that organisms must cope with through various strategies. Some animals migrate to regions where conditions remain suitable year-round. Some enter dormancy states, such as hibernation or estivation, where metabolic rate drops dramatically and energy expenditure is minimized. Some simply endure harsh conditions through physiological tolerance and behavioral adjustments, like reducing activity or seeking sheltered microhabitats. Plants may die back to underground structures, drop leaves to reduce water loss, or produce seeds that remain dormant until conditions improve. Disturbances such as fires, storms, floods, and droughts create patchwork landscapes where different areas are at different stages of recovery. Immediately following disturbance, pioneer species, tolerant of harsh conditions and capable of rapid colonization, establish in disturbed areas. Over time, competitive displacement and facilitation processes cause gradual species turnover as communities progress through successional stages. Eventually, late successional species that grow slowly but tolerate shade and competition may dominate until the next disturbance event resets the cycle in that location. These dynamics create spatial heterogeneity in community composition across landscapes. With different patches in different successional stages, depending on their disturbance history. This heterogeneity affects overall biodiversity, with some species specializing in early successional habitats and others requiring late successional conditions. The landscape scale diversity results from the mosaic of different local conditions created by the spatially and temporally variable disturbance regime. Genetic variation within populations provides the raw material for evolutionary change. This variation arises through mutation, which introduces random changes to DNA sequences during replication or through damage from radiation or chemical agents. Most mutations are neutral or deleterious, having no effect on fitness or reducing fitness. Occasionally, a mutation improves fitness in the current environment, and natural selection will tend to increase its frequency over generations. The accumulation of beneficial mutations over very long time periods can produce substantial evolutionary change. The rate of evolutionary change depends on generation time, population size, strength of selection, and availability of genetic variation. Species with short generation times can evolve more rapidly than species with long generation times, because more generations of selection occur per unit time. Large populations maintain more genetic variation and produce more mutations per generation, providing more raw material for selection. Strong selection, where fitness differences between genotypes are large, produces faster evolutionary change than weak selection. Limited genetic variation constrains evolutionary response, regardless of selection strength. Coevolution occurs when two or more species reciprocally influence each other's evolution through their ecological interactions. Predator-prey coevolution may produce evolutionary arms races, where prey evolve better defenses and predators evolve better offense in ongoing cycles. Plant-pollinator coevolution may produce increasing specialization, with flower morphology matched to pollinator morphology and behavior. Parasite-host coevolution may produce cycles where hosts evolve resistance to common parasite genotypes, and parasites evolve ability to overcome common host defenses. These coevolutionary dynamics can be modeled mathematically using population genetics and game theory frameworks. The models predict cycles, stable polymorphisms, or directional evolution, depending on the specific costs and benefits of different trait values and the genetic architecture underlying the traits. Empirical studies have documented coevolutionary dynamics in various systems, including bacteriophage interactions, plant-herbivore interactions, and host-parasite interactions in natural and laboratory populations. Biogeographic patterns in species distribution reflect both current environmental conditions and historical processes, including dispersal, vicariance, and extinction. Species occur where suitable environmental conditions exist and where they have been able to reach through dispersal, or where they have persisted following historical range contractions. Island biotas tend to have lower diversity than mainland areas of similar size because islands are harder to reach and support smaller populations, more vulnerable to extinction. Mountain ranges create barriers to dispersal and generate environmental heterogeneity, supporting diverse, specialized species. The latitudinal diversity gradient, with higher species richness in tropical regions than temperate or polar regions, results from multiple interacting factors. Tropical regions receive more solar energy, supporting higher productivity and more complex food webs. Tropical regions have been climatically stable for longer periods, allowing more evolutionary time for speciation. Tropical regions have larger geographic areas at similar latitudes due to Earth's geometry. Extinction rates may be lower in tropical regions due to milder climate. The relative importance of these mechanisms continues to be investigated. Phylogenetic relationships among species reveal evolutionary history and allow inference of ancestral states and trait evolution. Organisms sharing recent, common ancestors are more similar, genetically and phenotypically, than organisms whose common ancestor lived in the distant past. By comparing DNA sequences across species and using molecular clock techniques, scientists can estimate divergence times and reconstruct evolutionary trees, showing patterns of descent with modification over millions of years. These phylogenetic patterns demonstrate that all life on Earth shares common ancestry. Descending from ancient microbial lineages that arose billions of years ago when conditions on Earth first became suitable for self-replicating chemical systems. The earliest life forms were simple, single-celled organisms lacking nuclei or other complex internal structures. Over enormous spans of time, some of these simple cells evolved more complex features through processes involving gene duplication, horizontal gene transfer, symbiosis, and other mechanisms that increase genetic and cellular complexity. Approximately 2 billion years ago, a major evolutionary transition occurred when certain bacteria were engulfed by other cells, and became permanently integrated as mitochondria, the energy-producing organelles found in eukaryotic cells. This endosymbiotic event enabled much higher energy production per cell, facilitating evolution of larger cell sizes and more complex cellular structures. Later, another endosymbiotic event incorporated photosynthetic bacteria as chloroplasts in the lineage leading to plants and algae. These major transitions occurred not through planning or purpose, but through accidental cellular interactions that happened to produce stable associations with mutual benefits. Multicellularity evolved independently multiple times in different lineages, with cells remaining attached after division and gradually evolving specialization into different cell types, performing different functions. In early multicellular organisms, all cells were relatively similar and could perform most necessary functions. Over time, natural selection favored increasingly sophisticated division of labor, with specialized cell types dedicated to specific tasks like nutrient absorption, structural support, reproduction, or sensory detection. This specialization increased efficiency, but also increased interdependence, with individual cells no longer capable of independent survival. The evolution of complex body plans involved development of regulatory genes, controlling spatial and temporal patterns of gene expression during embryonic development. Slight modifications to these developmental programs could produce substantial changes in adult morphology, allowing evolution of novel structures and body organizations. Eyes evolved independently in multiple lineages through different developmental pathways, all converging on similar functional designs, because the physics of image formation constrained possible solutions. Wings evolved independently in insects, pterosaurs, birds, and bats through modifications of different ancestral structures, but all solving similar aerodynamic challenges. Sexual reproduction, found in the vast majority of animal and plant species, involves production of haploid gametes through meiosis and fusion of gametes from two parents to produce diploid offspring. This process generates genetic variation in offspring through recombination during meiosis and through combination of genetic material from two parents. Sexual reproduction is costly compared to asexual reproduction, requiring energy and time to find mates, produce specialized reproductive structures, and in many cases, provide parental care. Despite these costs, sexual reproduction predominates, suggesting significant benefits that outweigh the costs. The primary benefit of sexual reproduction appears to be generation of genetic variation that helps populations respond to changing environments and resist parasites. In constant environments, asexual reproduction would be more efficient, allowing rapid population growth without the costs of mate finding and sexual reproduction. However, environments change continuously due to climate variation, introduction of new species, and especially due to coevolution with parasites that evolve rapidly to overcome host defenses. Sexual reproduction shuffles genetic combinations each generation, potentially producing offspring with novel resistance combinations that parasites have not yet adapted to overcome. Parasites, because they typically have shorter generation times than their hosts, can evolve more rapidly and potentially adapt to overcome host defenses within relatively few host generations. This creates strong selection pressure for hosts to continuously generate genetic novelty through sexual reproduction. Mathematical models of host-parasite coevolution show that sexual reproduction can be maintained by this red queen dynamic, where hosts must continuously evolve just to maintain current fitness levels as parasites evolve to exploit them more effectively. Senescence, the gradual deterioration of physiological function with age, occurs in most organisms and leads eventually to death, even in the absence of disease or injury. Senescence appears paradoxical from an adaptive perspective, since natural selection should favor maintaining functionality as long as possible to maximize reproductive output. However, the evolutionary theory of senescence explains this phenomenon through declining force of natural selection with age. Mutations that cause harmful effects late in life are subject to weak selection because most individuals in natural populations die from external causes before reaching old age. So there is little selective pressure to eliminate late-acting, deleterious mutations. Additionally, mutations that provide benefits early in life, even at the cost of harm later in life, can be favored by natural selection if the early benefits outweigh the later costs when appropriately discounted by probability of surviving to later ages. This antagonistic pleiotropy can lead to evolution of traits that increase early reproductive success, but cause senescence later. The allocation of resources to reproduction rather than somatic maintenance represents another mechanism producing senescence, with organisms trading investment in long-term survival for increased current reproduction. In natural populations, senescence is rarely observed because external mortality sources like predation, disease, and starvation kill most individuals before senescence becomes evident. Laboratory studies removing external mortality sources reveal senescence patterns that would not normally be expressed in nature. The lifespans observed in protected laboratory or captive conditions typically greatly exceed lifespans in natural populations, demonstrating that intrinsic senescence limits are rarely reached under natural conditions. Immunosenescence, the decline in immune system function with age, increases vulnerability to pathogens and parasites in older individuals. The thymus, an organ involved in T cell development, begins shrinking after puberty in mammals, leading to decreased production of new T cells. Existing immune cells become less responsive to antigens and less efficient at eliminating pathogens. Chronic low-level inflammation increases with age, potentially causing tissue damage and contributing to various age-related pathologies. These changes result from the declining selection pressure for maintaining immune function at advanced ages that are rarely reached in natural populations. Telomeres, repetitive DNA sequences protecting chromosome ends, shorten with each cell division in most somatic cells. After sufficient divisions, telomeres become critically short, triggering cellular senescence or death. This replicative limit, called the Hayflick limit, restricts the number of times cells can divide. Telomere shortening may have evolved as a tumor suppression mechanism, preventing unlimited cell division that could lead to cancer. However, this protection comes at the cost of limiting tissue regeneration and repair capacity, contributing to organismal senescence. Cancer represents a breakdown in normal cellular regulation, with cells escaping growth control mechanisms and proliferating without the constraints that maintain organized tissue structure. Cancer cells accumulate mutations that disable growth inhibition pathways, activate growth promotion pathways, prevent apoptosis, enable unlimited replication through telomerase activation, promote angiogenesis to provide blood supply, and eventually allow invasion and metastasis to distant tissues. These changes transform normal cells into malignant cells capable of unlimited proliferation. From an evolutionary perspective, cancer represents somatic evolution occurring within individual organisms. Cancer cells are subject to natural selection within the body environment, with mutations that enhance growth, survival, or resource acquisition, increasing in frequency. This within-organism evolution can be remarkably rapid, producing genetically diverse cancer cell populations that may evolve resistance to treatments, just as microbial populations evolve antibiotic resistance. The body's immune system and other cancer suppression mechanisms represent selection pressures, opposing cancer cell proliferation, creating an evolutionary arms race within the individual. Multicellular organisms have evolved numerous mechanisms suppressing cancer, including DNA damage checkpoints that halt cell division when genetic damage is detected, apoptosis pathways that kill cells with severe damage, contact inhibition preventing excessive cell division when cells are densely packed, and immune surveillance eliminating abnormal cells. Despite these defenses, cancer remains common, especially at ages beyond typical lifespans, in natural populations where selection pressure to maintain cancer suppression mechanisms declines. The somatic mutation theory of aging proposes that accumulation of mutations in non-reproductive cells over an organism's lifetime contributes to functional decline and senescence. DNA damage occurs continuously from background radiation, reactive oxygen species, generated during normal metabolism, errors during DNA replication, and various environmental mutagens. Most damage is repaired by cellular DNA repair mechanisms, but some escapes repair and becomes fixed as mutations. Over time, mutations accumulate in somatic cells, potentially disrupting cellular function and contributing to tissue deterioration. Mitochondria are particularly vulnerable to mutation accumulation because mitochondrial DNA lacks the sophisticated repair mechanisms available for nuclear DNA. And because mitochondria are exposed to high levels of reactive oxygen species generated during oxidative phosphorylation, mutations in mitochondrial genes can impair energy production, further increasing oxidative stress and creating a positive feedback loop, accelerating mitochondrial deterioration. This mitochondrial theory of aging suggests that progressive mitochondrial dysfunction contributes to senescence and age-related pathologies. Free radicals, highly reactive molecules with unpaired electrons, are produced during normal metabolism, especially in mitochondria during oxidative phosphorylation. These radicals can damage proteins, lipids, and nucleic acids, potentially contributing to aging and disease. Organisms have evolved antioxidant defense systems, including enzymes like superoxide dismutase and catalase, and small molecules like glutathione and vitamin E that neutralize free radicals before they cause damage. However, these defenses are imperfect, and some oxidative damage accumulates over time. The free radical theory of aging proposed that progressive oxidative damage drives senescence, and that enhanced antioxidant defenses might slow aging. However, experimental tests of this hypothesis have produced mixed results, with some studies showing no effect or even negative effects of increased antioxidant levels on longevity. This suggests that oxidative damage is one factor among many contributing to aging, rather than the primary cause, and that the relationship between oxidative stress and aging is more complex than initially theorized. Developmental processes proceed through precisely orchestrated sequences of gene activation, cell division, cell differentiation, and morphogenesis, transforming a single fertilized egg into a complex, multicellular organism. These processes are controlled by regulatory networks involving transcription factors, signaling molecules, and epigenetic modifications. that determine which genes are expressed in which cells at which developmental stages. Small changes in these developmental programs can produce large changes in adult phenotype, providing a mechanism for rapid evolutionary change. Heterochrony, changes in the timing or rate of developmental processes, can produce novel morphologies through relatively simple genetic modifications. Pedimorphosis, the retention of juvenile features into adulthood, occurs in various lineages, including salamanders that retain larval characteristics and reach sexual maturity without metamorphosing into adult form. Paramorphosis, extension of development beyond ancestral patterns, can produce exaggerated features like the elaborate antlers of some deer species. These timing changes represent a mechanism for generating morphological diversity through modifications of existing developmental programs rather than evolution of entirely new programs. Phenotypic plasticity, the ability of a single genotype to produce different phenotypes in different environments, is widespread in nature. Many organisms adjust their morphology, physiology, or behavior in response to environmental conditions during development or even in adulthood. Plants grow taller in shaded environments, allocating resources to stem elongation to compete for light access. Many fish and reptiles have temperature-dependent sex determination, with offspring sex determined by incubation temperature rather than genetic factors. Some organisms develop distinct morphological forms in response to predator presence, such as zooplankton that grow defensive structures when chemical signals indicate predator presence. This plasticity can be adaptive, meaning the phenotype produced in each environment increases fitness in that environment relative to alternative phenotypes. Adaptive plasticity allows organisms to cope with environmental variation without genetic change, potentially buffering populations against selection and slowing evolutionary responses. However, plasticity itself is a genetically based trait subject to natural selection, and populations can evolve increased or decreased plasticity depending on environmental predictability and the costs of maintaining plastic responses. Reaction norms, graphical representations of phenotypic plasticity, showing how phenotype varies with environment for a given genotype, can differ among genotypes, creating genotype by environment interactions. When different genotypes have different optimal environments, no single genotype is universally best, and genetic variation can be maintained by environmental heterogeneity. This represents one mechanism maintaining genetic diversity in natural populations, despite natural selection's tendency to eliminate variation by favoring optimal genotypes. Genetic drift, random changes in allele frequencies due to finite population size, operates alongside natural selection as an evolutionary force. In small populations, random sampling of gametes each generation can cause substantial frequency changes for neutral alleles that do not affect fitness. Even slightly beneficial or deleterious alleles can increase or decrease in frequency due to drift. And in very small populations, drift can overpower weak selection. Over long time periods, drift leads to random fixation or loss of neutral alleles, with the rate of fixation inversely proportional to population size. Population bottlenecks, severe reductions in population size, cause loss of genetic variation through drift. As only a small sample of the original population's genetic diversity persists through the bottleneck, this lost variation cannot be quickly regenerated, since mutation produces new variation slowly. Bottlenecked populations may have reduced fitness due to loss of adaptive variation and increased frequency of deleterious alleles through drift and inbreeding. Many endangered species have experienced bottlenecks, reducing their genetic diversity and potentially compromising their evolutionary potential to respond to environmental changes. Inbreeding, mating between relatives, increases homozygosity and can expose recessive deleterious alleles that are normally masked in heterozygotes. Inbreeding depression, the reduction in fitness observed in inbred offspring, results from increased expression of these deleterious recessive alleles and from loss of heterozygote advantage at loci where heterozygotes have higher fitness than either homozygote. Small populations inevitably experience some inbreeding simply because all individuals are somewhat related, and this can create a positive feedback loop where small population size causes inbreeding depression, further reducing population size and increasing inbreeding in subsequent generations. Mutation selection balance describes equilibrium allele frequencies for deleterious mutations, where the rate of new mutations introducing harmful alleles balances the rate at which selection eliminates those alleles. For recessive deleterious alleles that are largely hidden from selection in heterozygotes, equilibrium frequencies can be relatively high, creating a genetic load where most individuals carry multiple harmful recessive alleles. For dominant deleterious alleles that are exposed to selection immediately, equilibrium frequencies are very low. The total genetic load from all deleterious mutations represents a continuous fitness cost that all populations bear. Gene flow, the movement of alleles between populations through migration and reproduction, homogenizes allele frequencies across connected populations and can introduce genetic variation into small or isolated populations. High gene flow prevents population differentiation, even when populations experience different selection pressures. While low gene flow allows populations to evolve independently. The balance between gene flow and local selection determines whether populations remain genetically similar or diverge evolutionarily. Metapopulation dynamics, the dynamics of multiple local populations connected by dispersal, create complex spatial patterns in species abundance and genetic structure. Some local populations may go extinct due to local environmental fluctuations or demographic stochasticity, while empty habitat patches are recolonized by dispersers from persisting populations. This extinction recolonization dynamic maintains regional persistence even when individual local populations are unstable. Genetic diversity is partitioned among local populations, with some alleles present in some populations, but not others. And dispersal redistributes this diversity across the landscape. Source sink dynamics occur when high-quality habitat patches produce surplus individuals that disperse to lower quality patches where reproduction cannot balance mortality. Without immigration from source populations, sink populations would decline to extinction. Yet sink populations can remain occupied indefinitely through continuous immigration. This creates misleading patterns where organisms are observed using habitats that cannot sustain viable populations. And removing source populations would cause secondary extinction of sink populations despite their continued occupation of apparently suitable habitat. Ali effects positive relationships between population density and individual fitness at low densities create additional extinction risk for small populations. Mechanisms producing Ali effects include difficulty finding mates at low density, reduced cooperative hunting or defense efficiency, increased predation risk. When group size provides protection, and inbreeding depression in small isolated populations. When Ali effects are strong, populations below a critical threshold density decline to extinction even in suitable habitat, creating an extinction vortex from which recovery is impossible without outside intervention. Habitat fragmentation, the breaking apart of continuous habitat into smaller isolated patches, affects populations through reduced total habitat area, increased edge effects, barriers to dispersal between fragments, and changes in predator-prey dynamics. Small habitat fragments support smaller populations more vulnerable to extinction from demographic stochasticity, environmental variation, and genetic factors. Fragments have proportionally more edge habitat influenced by conditions in surrounding matrix, potentially degrading habitat quality. Dispersal between fragments may be restricted by inhospitable matrix habitat, reducing gene flow and recolonization potential. Edge effects include altered microclimate conditions, like increased temperature, decreased humidity, and increased wind exposure near fragment boundaries. Light levels may increase near edges, where adjacent habitats are more open, favoring different plant species than fragment interiors. Predators and parasites may be more abundant near edges, where they exploit resources in both fragment and matrix habitats. These edge effects penetrate some distance into fragments, and in small fragments, edge-affected habitat may comprise most of the total area, fundamentally altering community composition relative to interior conditions. Corridors, strips of habitat connecting isolated fragments, can facilitate dispersal between fragments, increasing gene flow and recolonization probability. However, corridors also potentially facilitate spread of diseases, invasive species, and predators, and their net effect on population persistence depends on the balance between these positive and negative consequences. Empirical studies of corridor effectiveness have produced mixed results, with some studies showing clear benefits and others showing no effect or even negative effects, depending on species and landscape context. Climate change alters species distributions through shifting temperature and precipitation patterns, changing disturbance regimes, and modifying biotic interactions. Species track changing climate by shifting their geographic ranges poleward or to higher elevations as suitable climate zones move. However, dispersal barriers, habitat fragmentation, and evolutionary constraints may prevent species from tracking climate rapidly enough, creating mismatches between species distributions and suitable climate space. Species unable to shift ranges or adapt evolutionarily face increased extinction risk. Phenological shifts, changes in seasonal timing of life cycle events like flowering, breeding, or migration represent one response to changing climate. Earlier springs and warmer temperatures advance timing of many events. But different species respond at different rates, potentially disrupting ecological synchrony between interacting species. If a bird species times breeding to coincide with peak caterpillar abundance, but caterpillars advance their phenology faster than birds advance breeding phenology, breeding success declines due to phenological mismatch. These mismatches create novel selection pressures, potentially driving rapid evolutionary change. Ocean acidification, the decrease in ocean pH resulting from absorption of atmospheric carbon dioxide, affects marine organisms, particularly those producing calcium carbonate shells or skeletons. Lower pH reduces carbonate ion concentration, making shell formation more energetically expensive, and causing dissolution of existing shells under severe acidification. Corals, mollusks, echinoderms, and various planktonic organisms face challenges from acidification, with cascading effects on marine food webs and ecosystem function. The rate of current pH change exceeds rates during most past geological periods, potentially outpacing evolutionary adaptation capacity. Invasive species, organisms introduced to regions outside their native range, often through human activities, can dramatically alter recipient ecosystems through predation on native species, competition with native species, disease transmission, habitat modification, and disruption of ecological interactions. Invasive predators on oceanic islands have driven numerous bird species to extinction through predation on eggs and flightless adults that evolved without mammalian predators. Invasive plants can alter fire regimes, nutrient cycling, and habitat structure, fundamentally transforming ecosystems. Invasive pathogens have caused widespread mortality in native plant and animal populations, lacking evolved resistance. The ecological impacts of invasive species vary enormously, depending on characteristics of both the invading species and the recipient ecosystem. Some invasions cause dramatic immediate impacts, while others initially appear benign but produce subtle long-term changes in ecosystem processes. Some ecosystems resist invasion effectively, while others are highly invasible, though the factors determining invasibility remain incompletely understood. Simple species richness does not reliably predict invasion resistance, and invaded communities can be species-rich or species-poor. Trophic cascades occur when predators suppress herbivore populations, indirectly benefiting plants by reducing herbivory pressure. Removing top predators can trigger cascading effects through food webs, with increased herbivore abundance leading to increased plant consumption and altered plant community composition. Classic examples include sea otter predation on sea urchins, indirectly protecting kelp forests, and wolf predation on elk, indirectly benefiting streamside vegetation. However, trophic cascades are not universal, and many ecosystems show weak or inconsistent cascading effects depending on food web complexity and environmental context. Keystone species, species whose impact on community structure exceeds their numerical abundance, can have disproportionate effects on ecosystems. Beavers modify stream morphology through dam building, creating wetland habitat used by numerous other species. Some predators may preferentially consume competitive dominance, allowing inferior competitors to coexist and maintaining diversity. Some pollinators may be essential for reproduction of multiple plant species, with their loss causing cascading plant extinctions. However, the keystone species concept has been applied inconsistently, and identifying true keystones requires experimental evidence rather than assumption. Ecosystem engineers organisms that physically modify environments in ways that affect resource availability for other species, create heterogeneous landscapes supporting diverse communities. Coral reefs, constructed by coral colonies, provide substrate and shelter for enormous diversity of marine life. Termite mounds alter soil properties and vegetation structure across tropical savannas. Tree falls create canopy gaps in forests, initiating succession and creating spatial complexity in age structure and species composition. These engineering effects persist beyond the engineer's lifetime, creating legacy effects that structure ecosystems over extended time periods. Nutrient limitation constrains productivity in most ecosystems, with nitrogen limitation common in many terrestrial systems, and phosphorus limitation common in freshwater systems. Adding limiting nutrients experimentally increases primary productivity, demonstrating bottom-up control of ecosystem function through resource availability. However, increasing productivity at lower trophic levels does not necessarily propagate through food webs if top-down control by predators or herbivores regulates abundance at intermediate levels. The relative importance of bottom-up versus top-down control varies among ecosystems and can shift over time or space. Decomposition rates control nutrient cycling and determine how rapidly nutrients locked in dead organic matter become available for plant uptake. Decomposition proceeds through physical fragmentation by detritivores and chemical breakdown by bacteria and fungi. Rates are influenced by temperature, moisture, oxygen availability, substrate quality, and decomposer community composition. Slow decomposition causes accumulation of organic matter and nutrients in unavailable forms, reducing productivity. Rapid decomposition quickly releases nutrients, but also makes them vulnerable to loss through leaching or volatilization before plant uptake occurs. Stoichiometry, the relative proportions of elements in organisms and environments, constrains ecological interactions. All organisms require multiple elements in specific ratios, and mismatch between consumer requirements and food quality can limit growth and reproduction. Herbivores consuming plants with low nitrogen content relative to carbon must process large quantities of plant material to obtain sufficient nitrogen. This creates nitrogen limitation even when total food quantity is abundant. Imbalances in other elements like phosphorus or essential minerals similarly constrain consumer nutrition and population dynamics. Primary productivity, the rate at which energy is captured by autotrophs through photosynthesis or chemosynthesis, sets the foundation for energy flow through ecosystems. Global primary productivity patterns reflect climate gradients, with highest productivity in warm-wet regions like tropical rainforests and lowest productivity in cold or dry regions like deserts and polar areas. Within this broad pattern, local productivity varies, with nutrient availability, disturbance regime, and community composition. Total global primary productivity has likely increased in recent decades due to elevated atmospheric carbon dioxide and nitrogen deposition, though regional patterns are complex. Secondary productivity, production of consumer biomass, is constrained by energy transfer efficiency between trophic levels. Typically, only about 10% of energy consumed at one level is incorporated into biomass at the next level. The remainder is lost to metabolism, excretion, and inefficient conversion. This low transfer efficiency limits the number of trophic levels sustainable in ecosystems and creates biomass pyramids with much greater biomass at lower than higher trophic levels. Exceptions occur in aquatic systems where phytoplankton generation times are short relative to zooplankton generation times. This creates inverted biomass pyramids, where consumer biomass temporarily exceeds producer biomass. The processes that sustain biological systems continue operating at every scale, from molecular interactions within cells to global biogeochemical cycles. They proceed without awareness, without purpose, and without ultimate direction beyond the immediate physical and chemical gradients that drive them. Each process can be understood through scientific investigation as patterns of matter and energy transformation following physical law. And now, as the natural world settles into its quiet cycles, as forests, oceans, and stone continue without urgency, there is nowhere anything needs to change, nothing more to unfold, nothing to resist. The night in nature is not empty, it is simply inactive, processes slowing, energy conserved, systems resting. Let the darkness remain neutral, neither kind nor unkind, just present, just ordinary. Let the unseen continue its silent work, roots growing, cells dividing, organisms breathing without attention. Your body follows the same rules, slowing, reducing effort, releasing activity. Thoughts may thin out, not disappearing, just becoming less active, less demanding. Breath continues on its own, steady, unforced, unremarkable. As ecosystems persist through night, you may also remain still, doing nothing, needing nothing. Sleep is simply a phase, like darkness, like dormancy, like rest in the wild. There is no message, no meaning to uncover, only continuation. And as nature remains indifferent and stable, you may drift without effort into sleep. Not as an ending, not as a beginning, just a pause within an ongoing system. Good night and rest within the quiet of the natural world.